and today we're going to talk about a very important process happening in your cells constantly. Protein synthesis. Without protein synthesis, the only way you would get protein is by eating it. Well, we'd be eating more beans and meat than if protein synthesis were here. So, we've got some models. It's the comeback of the matchstick models. Here, we have a twisted double helix strand of DNA. In DNA replication, the DNA is purposely untwisted and then transcripted. But what happens when the DNA is transcripted? Well, when the DNA gets transcripted, the enzyme starts splitting this DNA up. And then you get this strand of RNA. How does this strand form? From RNA bases. Free RNA bases in the nucleus come and pair with these DNAs to get exactly the right RNA codon. The point of protein synthesis is knowing how does protein form. By the way, this is a messenger RNA, one of the only 3 types of RNA that exist in the human body. Unless you're getting infected by a virus, in which case there is genetic RNA material inside viruses that is what they inject into your cells to hijack your cells, make more, and you get sick. But for now, we have one of the 3 types of RNA, messenger RNA, but where does it go? See, DNA always remains inside the nucleus. It never gets out. But lucky for messenger RNA, it travels out of the nucleus. Out of the nucleus into the cytoplasm, which is the jelly-like substance where all the organelles are held. And this RNA moves across until it encounters a ribosome. This ribosome has RNA in it as well, called ribosomal RNA, the second of the 3 types of RNA in the human body. Now you may be wondering, what does this RNA do? A blue represents a thymine, a red represents a adenine, a yellow represents a cytosine, and an orange represents a guanine. Now, the green represents uracil, though that's only in this strand. So we have a strand TAC, AGC, TAC, CTG, TAC, TTT, and ACT, which is going to form the following strand. AUG, UCG, so AUG, GAC, AUG, AAA, and UGA. So those are the components in this RNA strand. What protein does this actually code for? Because the DNA codes for the proteins that you make. What does this code for? To help with that, we use what's called a codon chart. A codon chart contains every single possible combination of 3 bases of RNA and says which amino acid, which is the monomer of a protein, which amino acid does it code for. And the point of all this is that now the RNA will actually bind to the messenger RNA. The RNA goes, doesn't actually make a sound, but I'm doing it because, So the RNA scans the first molecule, and then the corresponding strand partner is sent over. Now, we have this amino acid, which is the one that codes for methionine. So this amino acid will code for methionine. And therefore, we will put a chain thing, because we are making a chain of amino acids. Well, better to do that after. So, now we have this tRNA. It goes off, does its business, and now we have another strand, and then the corresponding amino acid is sent. Okay, so far, so familiar. Oh, it's the second methionine. We have another methionine coming up. And then this continues. You look acid corresponding to it, and so on. The tRNA reads the code in here amino acid by amino acid until eventually you get to this base. Now this amino acid base doesn't code for an amino acid. There's no transfer RNA, which is the third type of RNA. Transfer RNA, tRNA, is the third of the three types. But there is no amino acid that this codes for. So what does this code for? It doesn't code for anything. This, in fact, tells you that the chain is over. This is the last component of any chain. Well, there are three different codons that when a ribosome scans it, it says that the amino acid chain is done. We have all the amino acids we need. Now, all we need to do is turn this into what we call a polypeptide chain. It's a chain of amino acids. This chain of amino acids can be pretty long, and that's its charm. But the amino acid chain is pretty long, and, well, it depends actually. In this case, it was only about 6 amino acids long, but in reality, they can be hundreds to thousands of amino acids long. So really, this is just the tip of the iceberg with a real protein chain. But what's up with this? Well, the thing is that you have this protein molecule, this polypeptide. This polypeptide is really just a chain of amino acids. The amino acid chain can get very complicated. Usually, there's some combination of all 20 amino acids in this chain. Now, we've made this amino acid chain, so it's like, okay, what now? So, well, this, my friends, is the protein that the cell is looking for. So the DNA, this DNA code codes for this specific protein, this specific chain of amino acids. The 4 different types of amino acids in here are methionine, serine, acylic acid, and lysine. So there are different types of amino acids in this chain, and that's what a protein is, a chain of different types of amino acids in a certain combination. That combination depends on the DNA strand. And since there are infinitely many DNA combinations, there are infinitely many possible different proteins. It's just most of them aren't very useful, and most of them aren't found in the human body. Sometimes organisms can have tens to hundreds of thousands of varying proteins. Unique. RNA is the star of this protein making. It's the star that made this protein synthesis happen anyway. Because what happened here is that the RNA scanned, and the transfer RNAs sent the corresponding amino acids and made a chain out of them. And this chain is in fact the protein that the cell is going to use. Why is this important? Well, it's because proteins are essential to life. They're building blocks. They can help protect against pathogens, build structures, and they are very helpful. In fact, without proteins, it would be hard to transport water from one place to another. And that's really important. Proteins are vital. So, the point of this is that protein synthesis is the way that we make the most important of the 4 biomolecules, protein. And the way those important stuff are made is worth knowing. Well, that's it for Making Math Easy, and I remind you to stay curious in math and science.